Well, welcome everybody. Thank you all for being here for the first of our summer webinar series of 2023. Today we'll be hearing from Dr. Richard Abbott, who has given us many webinars before in the past, and we're always appreciative to have him back to share his vast knowledge and expertise of botany with us. Today he'll be giving us a presentation on an introduction to plant phylogeny, understanding cladograms. We'll be going over part one, which is terminology and concepts. Dr. Abbott is an assistant professor of biology at the University of Arkansas at Monticello, where he is also the curator of the university's herbarium. We're very excited to have him back again this year. This webinar is being recorded. Afterwards, the recording will be uploaded to our YouTube channel. If you'd like to learn more about the Arkansas Native Plant Society, you can do so by visiting our website at anps.org. Joining the Native Plant Society is simple. Just go to anps.org/join, where you can use your PayPal account to join online, or there's also information on how you can send in a check to our treasurer to join that way. The next webinars we have coming up in our summer webinar series include Saturday, June 24th. That's next Saturday. We'll have Dr. Abbott back for part two of an introduction to plant phylogeny, understanding cladograms, in which he'll be giving us examples and case studies of various plant species and how things have maybe been moved around. But also Saturday, July 8th, 3 p.m., Karen Willard will be given a program on carex, species of Arkansas, carex being a very large genus of sedges. And then Saturday, August 19th, we'll have a 2-hour webinar starting at 10 a.m. Theo Witzel will be presenting on Natural Northwest Arkansas, the Benton and Washington County Natural Heritage Inventory, where we'll be talking about the results of the inventory that they conducted on the natural or the location and occurrence of different species of plants in Benton and Washington counties up here in Northwest part of Arkansas. All right, with that, I'll hand it over to Dr. Abbott. Doctor, thank you for being here.
Cool, thank you. All right, welcome all. And so essentially, as we've already heard, what we're here to talk about today is how to understand these cladograms. And so by cladograms, we're talking about these pictorial representations of hypotheses of relationships. Essentially, it's a way of visually showing what we think is related to what. What are the sister groups? Which groups are most closely related to each other? And that's fundamentally what cladograms are about. And so it ties into phylogeny. Phylogeny is a word that refers to the study of relationships. It refers to like a lineage birth. And so anytime you're talking about how things are related, that's just phylogeny, just means relationships basically. And cladograms are the pictorial way of representing those relationships. And so it can be quite complicated, but at the same time, it's not that hard once you understand the basics here, right? And so in the past, we had a couple of webinars about how to ID, and for me, that's what this cladistic stuff boils down to as well. And my passion is how to walk up to something and put a name on it. And it turns out if you understand cladistics, if you understand phylogeny, it shows you the patterns that are worth naming, the patterns that correspond to the morphology, right? And so whether you're talking about natural groups, and this is where this word clade comes in, clade is a synonym for a monophyletic group. Monophyletic meaning having a single origin. So mono, one, phyle referring to a branch. So a single lineage, a natural group is also called a clade. And so another way of saying it is based on the modern paradigm is this is how we define natural groups, monophyletic groups called clades, right? And so this is essentially what the modern paradigm is trying to do, is to make sure that all of our genera and family and orders and higher level groups are natural or monophyletic, are clades. And as we talked about in the past, that doesn't mean that artificial groups aren't useful sometimes. And so you can have non-monophyletic groups that are still diagnosable. So if the goal is practical plant ID, artificial groups can sometimes, again, be very practical, right? So we could talk about the baseline angiosperms and madcap rosy, these things we've talked about in the past, right? But the question we have is how do we go about determining which groups are clades and which groups are not monophyletic, or are not natural, basically, right? And so I want to just kind of reiterate that here today, the idea is to kind of go over this nutshell of what these changes mean for us, what this terminology means for us, and give a few examples. So it's essentially, once again, a tour into a handful of vocabulary words, and we're just going to spend the next hour each talking about this vocab and some of the conceptual framework here. If you have any questions, please feel free to post. I'll try to stop every now and then and ask for questions. But just one quick example of kind of what I mean by applying the cladistic stuff here is, here we're looking at a cladogram of a group called the Asteridae, the sympedally. If you walk up to a plant, it's a dicot, with the broad blades, the branching veins, and the non-sheathing base typically, and you find that the petals are fused. There's a 99% probability that what you're looking at is one of the families in the orders that are listed here, right? The squashes are the one kind of exception to that. There are a few monocots or sympedals as well. But essentially, this is the idea that we're talking about here, is using a cladogram to say, aha, it turns out that all these things with fused petals are each other's closest relatives. Well, when you start looking at the symmetry of the flowers, whether they're zygomorphic, also called bilateral or monosymmetric, right? These zygomorphic bilateral flowers characterize the lineages, which is the mint order, that includes the mint family and relatives, right? And so this is the idea of level of universality. At what point in time, at what point on the cladogram, which group are you talking about with this particular feature? That's the level of universality again. And so if you're talking about something that's sympedalous and bilateral or zygomorphic, that's the mint order. In contrast, if you're talking about something that's radial, has multiple planes of symmetry, is polysymmetric, and then has plications in the flower, actually has fold lines in the flower, that's the Solanales, the tomato order, which includes the tomatoes and the morning glories. Right? There's these thick multicellular glandular hairs called collators that characterize the Gentinales. The Gentinales are the family, is the order that gives us the coffee family, the milkweed family, things like this, right? And so it turns out that again, these individual features characterize specific groups. And when we're talking about a particular feature that characterizes a group, that's referred to as a shared derived feature or a synapomorphy. And then when you're talking about retained ancestral features, so for instance, the Lineales and Gentinales still have fused petals, but fused petals are retained ancestral features for those at that level of universality. So at that point, the fused petals becomes what we would then refer to as a retained ancestral feature or a plesiomorphy, right? And so once again, by understanding half a dozen basic terms here that are kind of complicated to spell sometimes, but by understanding them in the context, we're actually able to apply them in a practical way and put names on plants because of this. Now, of course, it's possible to just ignore all of this and just focus on a traditional approach to just memorizing families. You could do that, absolutely. You don't necessarily have to understand everything we're going to talk about here today in order to then be able to put names on all the families. It just helps simplify diversity if you can understand the patterns of relationship. And so, for instance, the tomato family that's discussed here is part of that tomato order, right? You could then just try to memorize all the features of the tomato family. So once again, what we're after here is kind of a practical way of using cladistics, to be able to talk about relationships, right? And so a clade, once again, is a monophyletic group. It's a natural group. A clade is a group that contains a common ancestor and all of its descendants. And so when we talk about cladistics, that's just another way of essentially talking about phylogeny. It's a way of talking about using patterns of relationship to understand change over time, to understand how groups are related evolutionarily, right? And so it's not exactly identical to human genealogy, because human genealogy are unrelated people marrying, right? But it's kind of like a family tree. You're tracking the evolutionary history of groups over time using these cladograms. So this is referred to as cladistics, which is just a particular methodological approach within phylogeny, which is the study of relationships. And so, for some reason, the words that we have here, basically, sorry, I went too many pages. These are the basic words that have to be understood in order to then talk about cladograms and understanding cladograms. And so phylogeny we've already defined as a study of relationships. Clade or cladogram. Clade is once again a natural group. It's a lineage. It's a monophyletic group, a group that has a common ancestor and all of the descendants. Cladogram is a pictorial representation of relationships. Monophyly means one lineage, one branch. It's essentially a fancy word that means a single lineage. Synapomorphy literally means shared derived features. And so these are the features that define groups. Plesiomorphy refers to like near features, or it's essentially referring to retained ancestral features. And we're going to come back and talk about all these repetitively here over the next few minutes. Homoplasy is a fancy word that just basically means evolutionary noise. When things are misleading us, and we're going to give a couple examples of this in a minute here. But homoplasy refers to false differences and false similarities. Essentially, when is morphology, or when is the evidence lying to us or misleading us? Basically, that's homoplasy. And then level of universality once again refers to the particular point in time, the place on a cladogram, or the group that for which a particular feature is relevant, right? And so, for instance, if we start off with humans, I think most of us probably accept that we're mammals, right? And so we have hair, but having hair is not unique to humans, right? So for humans, we're just a subset of mammals, having hair is a retained ancestral feature. It's a plesiomorphy. What's the level of universality for having hair? Is the group called mammals, right? All mammals have hair. Well, there's a couple exceptions, right, where they've lost the hair, but the bottom line is as a shared derived feature, as a synapomorphy, the level of universality, the correct group for that is called mammals, right? So humans also have legs. So we've got 2 arms and 2 legs. That's a retained ancestral feature. And having 2 arms and 2 legs, this is the group called tetrapods. This is here where you see the word legs on the cladogram here. Group called tetrapods. Well, it turns out that having the arms and legs is what lets us know that the amphibians and the reptiles and mammals are all related to each other. They're all characterized. This group called tetrapods is characterized by having the 4 limbs, right? Having the amniotic sac. Amphibians don't have an amniotic sac. There's this egg with an internal membrane that conserves water, right? Amphibians don't do that. Well, the reason we know that the reptiles are sisters of the mammals is because they have this shared derived feature, this shared synapomorphy of having an amniotic sac, right? So this idea that you can track relationships and then tease apart the similarity to talk about what is a retained ancestral feature, what is a shared derived feature based on the level of universality, and then not letting the homoplasy fool you. So for instance, birds, for the longest time, nobody thought that birds were reptiles. We now know that the birds are the last of the dinosaurs, right? That there were dinosaurs with feathers, that birds evolved out of the dinosaurs. However, for the longest time, people didn't accept that because birds didn't look like reptiles, or they had feathers. And so that's an example of a false difference, the same way that snakes are reptiles, and reptiles are tetrapods, right? But snakes have lost their arms and legs. So you can have false differences. Sometimes things look different, but are still the same, but you can also have convergence. So for instance, birds have wings. Well, so do bats, but bats are mammals, right? And so that's an example of false similarity. It's convergent. So once again, false similarities and false differences are homoplasy. Monophyly is the idea of a single lineage. So if you decided to talk about fish, for instance, and we've got the sharks and rays, the cartilaginous fishes, and then the bony fishes here. And then if you also threw in things like jellyfish, and then anything in the water is a fish, right? Fish are clearly not a monophyletic group. They're not a single lineage. There's several different lineages of things that we call fish, basically, right? So monophyly has become an important criterion for assessing, is the group that we're recognizing a single lineage or not? And so monophyly is one of the criteria that has to be kind of adhered to in the modern paradigm, right? And so these words, I'm not going to go through them all again here, but essentially this presentation is available as a PDF. So you can contact me or you can contact Eric. And you can go back through and study this, but essentially I've defined all these words already. They're here for you to go back and study on in your own time here. But I will stop and ask quickly here. Are there any questions at this point with all these words I've run through so quickly? Nope. Eric, no questions?
Don't see any in the chat, but folks, feel free to unmute your microphone if you have a question.
Cool. All right. Well, good. Hopefully it's because it was all so clear that you all understand what we're so, which is awesome. But basically, if you find yourself with any questions, please do follow up. And what we're going to do now is kind of move forward by providing a little more context for talking about this, and then we'll kind of reiterate some of it again. I will stress that there's a lot more to it. I've simplified, but by just having these few basic words here, I don't want to give you the false impression that it's not a lot more complicated than that, right? The truth is you could spend years and years studying all the additional nuances of phylogeny and cladistics. But what I'm trying to do here is just focus on the kind of a simple basic approach for the foundational grasp of what cladograms are. And so why most of us even begin to care about this kind of phylogeny is how it impacts our classification. We have decided in the modern paradigm that our classification should be linked to phylogeny. Not everyone agrees with that. I saw a treatment a few years ago where someone said, why should we let phylogeny influence our classification? Essentially, another way of wording that is classification can be artificial. It doesn't have to reflect relationships, right? But if you want to have the most predictive classification possible, if you want to have the best, most robustly supported here hypothesis, then base it on the data that we have, right? And so this idea that we can depict these evolutionary relationships using these phylogenetic trees, using cladograms, and again, it's telling us about sister groups. So if we looked at this here, this is a cladogram of carnivores, right? And if I ask you, what is the sister group to the weasel family? So the weasels include the badgers, the otters, martens, things like that, right? The minks. The sister group to the weasel family, the mustelid here, is actually the dog family. So to determine relatedness like this, you follow the branching point, the kind of the Y or the T that branches from that. And so the weasels here are sister to the dogs. And then collectively, the cats, the Felidae, are sister to the group that contains the weasels and the dogs. And so this is how you would interpret a cladogram, is that what we're saying here is, based on what we see here, we're saying that a wolf is sister to a coyote. We're saying that this genus Canis, the dog family, Canidae, is sister to the Mustelidae, the badgers and weasels, right? Based on the hypothesis here, we're seeing that river otter is sister to a badger, or also wolverines are in this group, right? Ultimately, not everything is here, so you have to keep something in mind when you're looking at a cladogram. It's very rare for a cladogram to actually be complete. So you have to understand that cladograms are these hypotheses of relationship, but they're only based on what's present in the actual analysis, right? And this ties into the idea of robustness. How well supported is that hypothesis? If the hypothesis is based on one small data point as opposed to 15 different data sets, right? How well supported is the hypothesis is called robustness. And so in a case like this pared down carnivore phylogeny here is very incomplete. The general pattern might hold up, but this is obviously not the true phylogeny of all carnivores, right? Because most carnivores are missing from this. So keep that in mind. Cladograms rarely show you everything, and you have to be aware of that. You have to keep in mind that it is not meant to be complete or thorough unless it's got a lot more taxes than this, right? The point here, though, is that this Linnaean system, the hierarchical classification, I think most of you are already familiar with, this idea of putting species into genera, into a genus, and then related genera into a family, related families into an order, and so on up, right? And we talked about this before from a practical ID perspective with plants. Anything above an order is, generally speaking, not very practical in the field. A couple exceptions like monocots, dicots, flowering plants, things like that. But generally speaking, this classification system that Linnaeus developed kind of pairs pretty well with a phylogenetic approach because you can have nested subgroups. I want us to keep in mind that each of these ranks, whether you're talking about a genus or a species or a family or an order, is called a taxon, plural is taxa, right? These different categories are taxa. And this is going to kind of be confusing perhaps, but rank is arbitrary. What defines a family, what defines a genus is a human construct. We're making these groups up. And the more informed we can be, the better informed our classification can be using phylogeny, phylogenetic understanding, the more robust, the better supported the hypothesis will be. But what it leads to as well is people quibbling over rank. So one person might say, well, this should be an order, and someone's like, no, that should be a subclass. Because there's all kinds of other ranks that can be intercalated here. You can have a subgenus or a subfamily or a superfamily or an infraorder. There's all these other levels that can be put in, right? And people will historically quibble over this. The modern paradigm is say, you know what, let's quit arguing about rank. Rank is arbitrary. Let's just think about the natural groups. Let's think about the clades, right? And so I avoid most taxonomic arguments these days by just saying, hey, I don't care what name you want to argue about. Let's just go ahead and think about the clades involved, right? And so essentially, but what I want to stress here again with that is if you start off with something like a species, like a bear, like the American black bear here, question comes, well, what's its closest relatives? Well, other bears, right? Other members of the same genus are going to be its closest relatives. Well, what are the closest relatives of the genus Ursus? Well, other genera related to bears, right? The family of the Ursidae, right? What are bears' closest relatives outside the bear family? Well, other carnivores, right? So you throw in the carnivores. So we've got this again, this kind of nested hierarchy going on here. And this reflects phylogeny, and our classification system dovetails very beautifully with the Linnaean hierarchy. There are exceptions. There are times when there are problems here, but we'll set those aside for a different time here. But understanding that when you're looking at a cladogram, what you're looking at is essentially a pictorial view of relationships across life. Right? You're looking at the ways in which we're saying these things are related to each other. That's what cladograms are. Very much like this classification system of saying that, well, all animals are more closely related to each other than they are to other plants, right? So it's the same thing. Cladograms are just pictorially representing relationships for us. One of the beauties of this kind of modern approach is that it's no longer a matter of personal opinion. I mean, historically, basically, there would be an appeal to authority. I would say, I'm the world's leading specialist with ducks, or I'm the world's leading specialist with oaks. And then you would either choose to believe me as your personal Lord and Savior in a biblical sense, right? You would just follow my beliefs without conscious thought of your own. Well, that's not happening anymore. Nowadays, you should be questioning everybody and everyone, because it's no longer just based on someone's personal opinion. It's no longer based on this kind of overall similarity or artificial uses or gross morphology. It's an attempt to look at a change over time and account for all the available data. What does the chemistry have to say? What does the floral anatomy have to say? What does everything about the plant have to tell us, right? And in order to understand why this is valuable and why this is meaningful, you do have to stop and remind yourself about evolution, basically. The reason why this turns out to work is that we've had this variable and changing Earth over the last billions of years, and that there's variation within individuals, meaning that some are more suited than others at particular points in time. So there's been kind of natural selection for certain types of morphology, right? But what leads to this variation among individuals? The genetics, the DNA. And I'm going to just touch on that very briefly here, just to point out that yes, most modern phylogenies are DNA based, and this leads to a problem because when you're in the field, you can't see DNA, right? So if we've got phylogenies based on DNA that don't match what we wanted the morphology to be, what are you going to believe? Your morphology or the underlying DNA? Well, this, and there's some conflict there, and then so admittedly, this is a problem sometimes. How do you resolve this conflict, right? But generally speaking, the more close related organisms are, the more similar the DNA is going to be. I think most of us can accept that if groups have, if two different organisms have similar DNA, it's probably because they're related, right? And that's underlying this approach. And so once again, I'm not going to repeat every bit of this, but this paradigm shift that has happened in the last 20, 30 years of using cladograms, this kind of cladistic approach, involves a transparent analysis of all the available data. It's no longer an appeal to authority. We're focusing on these unique shared derived features, the synapomorphies, not just overall similarity, but specifically what are the synapomorphies, the shared derived features. We're then using these cladograms to track evolutionary history. We're looking at descent modification over time and space. And we're also then going to focus on only naming the monophyletic groups, the groups that are actually natural, right? The groups that contain a common ancestor and all of its descendants. And so with this particular slide, I've kind of ended the everything that needed to be said in terms of vocabulary. But there's obviously several more slides to go here. I'm looking at that's 14. There's 30 something more slides to go. We've still got a good chunk of time. So we're gonna move on. But basically everything from here on is just kind of reiterating what we just talked about. So I wanted to stop again and ask if there were any questions at this point in time before we start reiterating. Nope. Okay. All right. So we'll move on then. So stepping back for a second here and remind ourselves of this idea of, okay, if this monophyletic thing matters, why is this a criterion, right? But it turns out that life had a single origin, right? Well, life is monophyletic. Life had a single origin. Life is a single lineage. As far as we can tell, life was started, or if you're a person of faith and life was created one time, that's fine. There's no conflict between the idea of creation and things changing over time, right? Ultimately, whatever you choose to believe is fine with me. What we know is that life had a single origin. All the evidence suggests to us that life had a single origin.
Hey, Richard, we got a question in the chat here. Shayna asks, is DNA analysis the reason things are being reclassified occasionally?
Yeah, so basically what it boils down to is, yes, is that DNA often tells a different story than morphology. And more importantly than that, it leads to showing that some of our groups were artificial, that we had a lack of monophyly. So the main reason the group names are changing, things being reclassified, is because the traditional groups were shown to be artificial, not natural. And the current paradigm insists that we have monophyletic groups. And so, yes, DNA is often the evidence that shows us that the traditional classification was artificial, but names are changing because we want to have natural groups, not just because DNA says so, but because we have a paradigm that insists on only naming natural groups. Hopefully that was clear enough.
A follow-up question here from, I'm sorry if I mispronounced your name, Gaurav. In addition to morphology and DNA analysis, does phytochemistry play a role in classification nowadays with the advent of chromatography techniques?
Yeah, so even before DNA started, a lot of the kind of earth-shaking new evidence was chemistry. Was essentially what was going on with the chemicals, and so there were people studying, like the development of the ovules and cellular level details, and studying secondary chemistry. The chemistry and the other molecular data that predated DNA was the beginning of what kind of ushered in the modern age. But absolutely everything is used. Everything. If we know something about the plants, we use it basically. We don't throw out any of the data. We don't throw out anything and say that's not important. Historically, they would have. Historically, they said, oh well, who cares about that? That's not important. Nope. If we know it, we put it into the analysis basically in the modern paradigm. Okay, cool.
Yeah, I think we can move forward. I'm not seeing any other questions at the moment.
Okay, cool, thanks. So we know that life is monophyletic. We know that life has a single origin because, for instance, all life uses the exact same DNA, right? If life had multiple origins, you would have expected that maybe different life forms would have had different types of DNA, right? We know that life is monophyletic because all life is cellular, right? There is no life that doesn't exist within a membrane-bound cell, basically, right? And all life uses the exact same energy molecule, ATP. So we've got multiple lines of evidence like this, which again, kind of microscopic, molecular, chemical things going on here that are telling us that life had a single origin. So no matter how different plants and animals are and how different they are from bacteria, they all share certain features in common. And so these features, these shared derived features of life, these synapomorphies of life, make it quite clear that life had a single origin, that life is a group that contains a common ancestor, life originated at one time, and then it's changed over time, right? And so that's the kind of the gist of this, is that okay, if we accept then that life is monophyletic, in fact we know that it is. The question then becomes, my screen's being blocked by all these. I don't know if y'all don't give presentations. There's all these little pop-up things that block the screen while I'm trying to talk. Makes it really confusing. I can't see half my screen from all the things in the way. But anyway, sorry about that. I can't read the slide right now. But we know that the groups have changed over time. We know that life started one time, and then various subgroups kind of changed evolved over time. This is the idea of the cladistic approach, right? Is let's track that change over time, and then let's map the relationships, right? And so when you stop and think about this idea that all life had a single origin here, and it's changed over time. This is why we find that 5% of our DNA is still identical to that of a bacterium, right? Why do humans? Why are 1,000 of our genes the same as bacteria? Well, those are the genes that cannot change. If you mutate those genes, life doesn't happen, right? Those 1,000 genes are the most important genes for life, basically, right? And they've never mutated. The other 20,000 plus genes in the human body have changed over time, but we still have 50% of the same DNA as bananas and 99% of the same DNA as chimpanzees, right? So this idea of using DNA as a proxy for how closely related things are makes a lot of sense, right? Because the more closely related you are to something, the more similar your DNA is, right? And so what this leads to then is this ability to then kind of link the phylogeny to a classification system. And so when you look at life, there's these three major domains, right? The bacteria and the archaeans. Historically, 20, 30 years ago, the archaeans were considered to be bacteria. They were just referred to as prokaryotes. But we now know that these archaeans are more closely related to the eukaryotes than they are to other bacteria. They've got some microscopic cellular details and chemical differences, right? And that the eukaryotes are the group that contain the animals, the plants, the fungi, the protists, right? And biology, of course, is the study of all life here. But essentially, this idea that we can start breaking up life into the different subgroups, and not just kind of in your mind carry this through. If we can talk about the natural subgroups within life, the natural subgroups within plants, the natural subgroups within this particular family, that's what our classification is trying to reflect. What are the natural subgroups, right? And so there's still a certain amount of arbitrariness about which groups to name. How do we decide which groups we're going to name, which groups we're going to talk about? That there's still a certain amount of arbitrariness there, right? That ultimately, and what I mean by that is if we jump forward here, I could just talk about group A, group B, group C, group D, group E, whatever, without talking about all the details in there, right? I could simplify this cladogram, make it look like I was only talking about 3 things or 4 or 5 things, right? At what point do you decide to name a group and talk about a group, to talk about a lineage? That's where the objectivity, that's where the subjectivity, that's where the objectivity is lost traditionally, right? Traditionally, it was just someone's personal opinion. And in the modern world, there's still a certain amount of subjectivity to this, but it's constrained by some of the objective rules and kind of regulations that we come up with, like a stress on monophyly and such, right? And so, for instance, within the eukaryotes, this clade called eukaryotes, we know that we've got the plants, animals, and fungi being discrete lineages, right? The synapomorphies of plants, so that they produce their own food. But that's kind of a weird thing because there are photosynthetic bacteria, right? So being able to photosynthesize, plants did not evolve photosynthesis. Bacteria did, and plants stole it from bacteria. So the synapomorphy of plants was having primary endosymbiosis with chlorophyll A, right? And another way of wording that is that plants acquired the ability to photosynthesize from bacteria. Plants are the only lineage that acquired the ability to photosynthesize directly from bacteria. That's what defines a plant, right? And so we can separate animals and fungi and plants based on words like that. And you can talk about these as synapomorphies if you find the right words. But then we're left with the remainder of the eukaryotes are not each other's closest relatives at all. These things called protists, right? Several different clades. So from a modern perspective, we no longer recognize a group called protists as in terms of our formal classification because it's artificial. And this is what we're getting at with the monophyly. If it's not a natural group, if it's an artificial group, and if you remember from before, we tend to put quotes around artificial groups. If it's an artificial group, it's not formally being recognized in the modern paradigm. So it's kind of circular here, but part of how we assess the artificialness or the naturalness of a group is by trying to interpret the homology of structure. So if you look, for instance, at the appendage of a human versus a cat versus a whale versus a bat here, you can track the different bones in the appendage there, right? Looking at similar structure and function and position and development, genetic control was historically how homology was assessed. Nowadays, homology is largely assessed just by looking at the cladogram. If 2 groups are not related to each other, then any similarity they have is not homologous. It's false similarity. And if 2 groups are similar, then it's assumed that there's homology there. But historically, even before the modern paradigm, homology was assessed with attempts at rigor, right? And so this is where we can once again now start talking about this convergent evolution, this idea that some similarity is not based on relatedness. Wings evolved multiple times in animals, right? Think about the insects that fly and the birds and the bats, right? They're not each other's closest relatives. We know they're not. And this kind of false similarity tells us nothing about relationships. Analogous similarity does not tell us about relationships. It might still be useful. So, for instance, imagine if you were a stranger to this planet and you just saw winged animals and you created a group of winged animals. That would be useful in your ignorance about the groups, right? It would be artificial. Well, in the same way, in the plant world, we have some artificial groups that are useful, but nonetheless, we don't formally recognize these taxonomically anymore, and that's why names have changed. Once a group has been shown to be artificial, we basically change the names to try to reflect the natural groupings. And so we can tease apart the overall similarity. We can tease apart the overall similarity. So trying to figure out how to there we go. We can still tease apart the similarity by once again saying, okay, that artificial convergence, we're not interested in that at all. That's just tells us nothing. So I used that word homoplays earlier, right? The idea of evolutionary noise, false differences and similarities. False similarities don't tell us about relationships, so we ignore that kind of similarity. Retained ancestry, what we call plesiomorphies, right? This is about level of universality. If you're talking about the level of universality of mammals, then having hair is a synapomorphy, right? But if you're talking about a group called humans, than having hair as a retained ancestral feature and doesn't tell us about relationships amongst humans, right? And so we're going to pull out these pleomorphies as well, these retained ancestral features as well, and say, yeah, they're important at a different level of universality. But if you're talking about a specific group where that feature is not a synapomorphy, then we ignore the pleomorphies, the retained ancestral features. And so the modern paradigm, once again, and again, part of why these groups have changed names and such, is that this focus on these shared derived features, understanding the specific level of universality, is allowing us to tease apart similarity, basically, right? And keep in mind as well the artificial differences that make up the other half of homoplasy, like the snakes and not having legs and arms, right? So essentially, that what we've learned to do is we've learned to better interpret artificial similarity and versus natural similarity, and we're now teasing all that apart, right? Come on. I'm not going to go into details. This is just here for people that want to read this. Most people treat these words as all been kind of more or less synonymous. But if you're a if you work with phylogenetics, if you're a taxonomist, if you're a systematist, if you work with nomenclature, we differentiate amongst these words because, for instance, if I wanted to put a name on something, if I had a plant, I didn't know what it was, I'm not going to go to a systematist. I'm not going to go to someone who works with phylogenetics. I'm going to go to a taxonomist because that's the person that studies the groups, right? But if I want to know about the naming of something and the rules, then I'm going to go to a nomenclaturist, right? If I want to know about, well, how do we know these things are related? Then I'm going to go to someone who works with cladistics, phylogenetics, right? So there are differences in these words, even though they all get used kind of interchangeably by some people. Kind of like there's different kinds of mechanics, different kinds of lawyers, right? So just be aware of that. And why I stress this is essentially you will find some people using words to mean slightly different things or using them in different contexts. So you have to know the kind of the mind for the mindset of the person that you're consulting, basically. Are they coming up from a systematic perspective or a taxonomic perspective or a nomenclatural perspective, right? So be aware that there are sometimes subtle differences out there, and you just kind of have to scratch the surface a little bit. Reiterating again, what is a clade? Just talking about this for a second here. This idea of kind of trying to stress again that a clade is a group that has been shown to contain a common ancestor and all the descendants. So if you look at this cladogram here, the simplified cladogram I've drawn here, this kind of graphic representation of relationships, right? What we're saying here in this cladogram here is that D is sister to E. The group E, which might be all monocots, or might be might be the oak family, or might be anything basically, right? It's not just a single thing. It might be, and A, B, and C might be the same. So when we're using these, keep in mind these might be entire groups, not just single organisms. But what this cladogram is showing us is that the group D is sister to group E. If you look right here, that V, that Y, where they come together, D is sister to E. C is then sister to the group DE. B is sister to the group CDE, and A is sister to the group BCDE. But if you try to recognize the group ABCD, that is not a monophyletic group because you're leaving out E, which is part of the same clade. Right? ABD would not be a monophyletic group, right? Because you're leaving out C and E. If you decide to recognize the group BE, that would be artificial. So again, names have changed because cladistic analyses, phylogenetic studies have shown that some of the groups that we're recognizing were artificial, were not monophyletic, right? So this is the fundamental basis as to why names have changed, is that modern cladistic analyses have shown that the groups were artificial. You can go into this even deeper and start talking about paraphyly versus polyphyly, and it's not an argument I want to get caught up in, because it's really about how inclusive you're being with those hypothetical common ancestors. I just prefer to talk about monophyly versus not monophyletic, right? But if you have a non-monophyletic group, it might be that it's because you've left out some of the descendants, or it might be because you're putting things together that are completely unrelated, right? And so once again, this is why names change, because we realize this. We realize that historically people were recognizing groups that were not natural, that were not monophyletic. One of the beautiful things about these cladograms is they can be used to track transformations. You can actually track the change over time. And so, for instance, what we're looking at here is a cladogram of the vascular plants, the land plants, showing how the angiosperms, the flowering plants, are sister to the gymnosperms, the conifers, right? And so where it says group C there, that would be the group that we would call seed plants. So this is the group that has seeds. And so that we drop down to B here when we've got the liverworts, which is a type of bryophyte, right? Sister to these other groups here. again, remember what I said about cladograms are leaving things out? Where are the ferns? Where are the fern allies? There's all kinds of things that have been left out of this cladogram here. This is not meant to be an absolute truth. It's simply showing us hypothetical relationships based on what's actually been mapped, right? But so nonetheless, we can still track change over time with the cladogram. But do keep in mind that if someone has not included everything they should have included, garbage in, garbage out, right? Sometimes when you look at a cladogram, it's going to be inherently false because it's just left too much out. So you have to always be aware of what's gone into the cladogram, what should have been in the cladogram that might be lacking. One thing that kind of confuses a lot of people too when they look at various cladograms is that nodes are reversible. And so these cladograms are all completely identical. They don't look identical. They look different, right? But if you think about them, when I say node, we're talking about the Ys or the Vs where the branches come together, right? Every one of these cladograms shows that D is sister to E, right? D is sister to E, D is sister to E, D is sister to E, D is sister to E. Every one shows that C is then sister to DE, right? C is sister to DE, C is sister to D in each of these. Every one of them then shows that A is sister to the group BCDE, right? These 4 cladograms show the exact same hypothesis of relationship. They're identical, even though they don't look identical, right? So that's the other tricky thing about cladograms is keeping in mind that the nodes can actually be reversed, and it doesn't change the pattern at all. It's about sister group relationships. So I'm going to wind down here by talking very briefly a little tiny bit about history, and then I'll end with 3 specific examples, kind of previewing for next time. But basically, these cladograms, this idea of family trees goes all the way back to the 1830s. Darwin, in his field notes in the 1830s, drew a cladogram, right? In fact, I've known several kind of like biology nerds, students who've gone out and gotten this as a tattoo. Basically, it's kind of a strange thing in my mind, but cladograms have been around for a long time. They just haven't really caught on as a methodological paradigm until fairly recently. It wasn't until the 1950s this guy Hennig, a German entomologist, started using cladograms in his publications, and he showed that cladograms can be used to represent these morphological shifts. He showed that cladograms can be used to represent the shift from tocogenesis to cladogenesis. And what I mean by tocogenesis is population level gene flow versus the formation of new lineages. So Hennig was showing, hey, you can look at this and you can actually track how populations are changing and shifting into species, which is a cool thing, right? He also showed that you can use cladograms to essentially track change over time between different groups. He showed that they can be used to represent nested hierarchical groups. Once again, these hypotheses of relationship. And this is in the 1950s. He showed that cladograms can be used to represent what's referred to as area cladograms. You can use them to track the movement of organisms across the planet, right? So there's all these uses for cladograms. And the question becomes, well, if he saw that back in the 1950s and Darwin was using it in the 1830s, what's the big deal now? Is that the paradigm has shifted to the point where this has now become a foundational part of what people have to do. But since the 1980s, people have been aware of this phylogenetic approach, but it generally takes decades for ideas to make it into the mainstream before the ideas get caught on. And people are pretty resistant to change, especially when that change is in another language, basically besides the one they speak, right? So Hennig's ideas finally caught on in the 1980s and are still pushing through today. Is there a question? I'm hearing some noise. Nope. Okay.
I think somebody's mic might have been unmuted. Elizabeth, did you have a question? Elizabeth Pollard?
Oh, no, I'm sorry.
Oh, that's okay.
Okay, cool. All right, thanks. So we're almost done here. We've just got a couple more things to wrap up on here. So this idea, though, that there's still a subjective element, that there is still a subjective element to which clades are worth naming, right? How do we decide which clades to put names on? Well, the primary criterion is monophyletic. When you've seen names changing in recent years, it's because some of the groups were not monophyletic. But the secondary criteria include things like stability, like tradition, minimizing redundancy. Some of us like to have groups that make identification easier. Other people say, nope, the identification has nothing to do with it. What's the predictive value? Are the groups diagnosable? What's the size of the groups? How old are they? So, for instance, a lot of the names that have changed in ferns in recent years have been because the fern lineages are very ancient, and people have just decided, oh, well, they're old lineages. We're gonna call it a new genus, a new family, just because it's old, right? Should that be happening? This is where the subjectivity is, right? And one of the things I wanted that we're not going to talk about as well, we'd need several more hours, but the whole idea of speciation. if you think about how species evolve, if you've studied that ever at all, speciation theory, most species should not be monophyletic. Species evolve out of other species, right? And so if genera are groups related species and the species started out as not monophyletic, why on earth would you expect all the genera to be monophyletic, right? And so there's definitely some room for discussion and argument here, but that's the context for a semester-long taxonomy class, right? And so we'll look at a few examples next time of this, but what I want to do now is wrap up by just showing 3 brief examples here. 1, about how birds, the last of the dinosaurs. 2, about how fungi were considered to be plants, but we now know they're sister to animals. And then 3, this group called angiosperms that were historically thought of as monocots and dicots, but we now know also need to have these kind of basal angiosperms recognized, right? And so next time we're going to talk about some specific examples as to why some of the names have changed. But for now, at least, example 1, here's a cladogram. And what we know is that birds evolved out of the Saurischian dinosaurs. In fact, the Saurischian dinosaurs, the ish refers to like hips. So you see the ish and the star referring to lizard. The ornith is a bird, right? The ornithischian dinosaurs literally means bird hip dinosaurs, right? Birds evolved out of dinosaurs. We know this now, right? And in fact, there are fossil dinosaurs where they've been found nesting with eggs and with feathers, right? So essentially, the dinosaurs did not go extinct. They're still with us as birds, right? And that's what this cladogram shows. This cladogram shows that relationship, that birds evolved out of dinosaurs. And so this is kind of the value, again, of this cladistic approach, is showing things like this. Fungi, traditionally seen as plants. Why? Well, because they don't move around, because they're attached to the soil. They have cell walls, right? Things like this, people said, oh, they must be plants of some sort. It turns out that if you look at modern phylogenies, though, fungi are sister to animals. And again, there's things being left out. The choanoflagellates aren't being talked about. There's always groups being left out. But if you look at the plants, they're green algae and plants, they're not closely related to fungi at all, right? They're in completely different lineages. And so the question becomes, well, how on earth do people call fungi plants? Well, again, we already talked about that. But what we know now, though, is how do we know that fungi are sister to animals? If you look at a table like this, fungi and animals both store their starch. They don't store starch. We store our carbohydrates as glycogen, right? In terms of the immediate energy use molecules, glycogen. Fungi store their carbs in the same way that animals do. And if you look at something called chitin, chitin is that chemical that makes up the exoskeleton of certain arthropods, right? Well, it turns out the cell walls of fungi are made of chitin. Chitin evolved one time in the ancestor to animals and fungi, and plant cell walls are made out of cellulose, right? So even though fungi were considered to be plants because they have cell walls, their cell walls are actually made of a chemical only otherwise found in animals, right? We know that fungi and animals are closely related. And example 3, the angiosperms we talked about, again, this is a fancy word that just means flowering plants or fruiting plants, right? The angiosperms historically were talked about as monocots and dicots. But now we have these several different unrelated lineages that I like to refer to as the basal angiosperms. Some people say primitive, but we try to avoid that. But these basal or early derived angiosperms that I conveniently dismiss by putting quotes around them as an artificial group have to be recognized. All the evidence tells us that these groups that were historically called dicots are not really actually dicots. The basal angiosperms were historically called dicots, but they're not dicots at all, right? There is a lineage called dicots. There's a lineage called monocots, and there's an artificial assemblage of early derived things that we're now kind of conveniently calling basal angiosperms, right? And so this is kind of what the cladogram has done for us. It's allowed us to provide a pictorial understanding of what all the data tell us, right? So dicots and monocots are still recognized, are still there, but we now also have to talk about these basal angiosperms, which include things like magnolias and pawpaws and water lilies and such, right? And so next time, basically, I have several different families that I could potentially talk about, but I wanted to. I just kind of wanted to kind of open this up to y'all's feedback here. Are there particular groups and questions that you have? Are there particular families or genera, particular cases of name changes that you would like me to address next time? Because that's what next a week from today we're going to talk about some specific examples of name changes, and I would love to address any specific questions y'all might have. So do you have any?
Feel free to unmute your mic if you would like to request anything from Dr. Abbott or ask a question.
And so I will go ahead and make it clear that even if y'all don't provide guidance on what I know, I'm going to talk about specific groups. I'm just willing to modify which groups I talk about based on those interests is what I was trying to basically offer. But I'm going to talk about like why the purslanes and cacti what happened to the Liliaceae, what happened to the maples and the Sapindaceae, what happened to the milkweeds, why did this corkwood, why did our corkwood become part of the Similarity? Things like this. I'm gonna talk about some of these examples next time, but I would also love to answer any specific issues that y'all might have.
Yeah, Shayna has requested Cornaceae and Nisaceae. I'm not sure how you say that, but I think it's the black gum cinnamon family.
Cool. Awesome. I'll make sure to talk about that one.
Okay. And then another question from Gaurav. Where do we place the aquatic / underwater, those found in oceans, plants in the classification, since we classically talk only about terrestrial plants?
Yeah, so kind of like with the animals, how we know that most of they're mammals in the ocean, right? The things like the dolphins and the whales are actually terrestrial animals that went back into the water. Almost all of the aquatic plants in the ocean are monocots that went back into the ocean. So essentially, these things like Zosteraceae, Poseidonacea, Rupiaceae, some of these things like this, they're actually different lineages of monocots, of aquatic monocots that went back into the ocean. And so we do talk about terrestrial plants, but those aquatic, underwater, oceanic plants are mostly monocots. And so they're mostly in the Elismatales. They're most related to things like Elismataceae and distant cousins to the Araceae and things like that. Cool. Well, so from my perspective, I mean, I was trying to keep this brief. So basically, I was done. I just wanted to let y'all know that this is next week. We're going to pick up where we left off here and talk about some specific examples of how and why names have changed over the last few decades.
Hi, this is John in Fort Smith. I'm interested. I'm not an expert in these areas. I'm a physician, actually. We're just kind of broadening my scientific view. I'm really interested in hearing you as you go into specific examples next week, talking about the data that you actually observe, and then at what point does the cladogram move into a hypothesis of a description, understanding that where does that balance point actually occur? Because at some point, the sister relationships are, as you said, a human hypothesis and not direct data. So I'm very interested, because we have the same issue in medicine where people talk about various concepts that aren't directly observed. And I like to be very precise in just defining what we observe, what is data and what is theoretical construct. So just as you think about preparing for next week, as you give your examples, I'm not as expert in all the genera here that were described, but that intellectual process of data and theoretical construct is of great interest. So I'd appreciate that.
Cool. Yeah, I can respond very quickly. I'm very brief to that right now as well. Basically saying that this is exactly part of the issue, that historically one person would have an opinion and they wouldn't have to explain what the source of the data was, right? So the beauty of this cladistic approach is that every single publication these days tells you exactly what the data are behind their hypothesis. And it's almost always just molecular data, almost always just DNA data, right? Which is kind of disappointing. But the most beautiful papers will actually tie in morphology, anatomy, chemistry. But it's very rare for someone to go to that level of detail. But ultimately, these hypotheses, these cladograms are always just hypotheses based on whatever limited data went into the analysis. And that's why they often end up being proven wrong later, because garbage in, garbage out. If someone doesn't do a complete, thorough, robust study, someone else might come along and do a better job and show why the previous person was wrong, right? But yeah, it's a fascinating thing, and it's a good thing to keep in mind. But generally speaking, if someone's done a DNA analysis, that's good enough for most people, and they don't worry about anything else these days.
We see similar phenomena in medical genetics. It's a very powerful technique, but it's not everything. Exactly. And in my work, I have to deal with real living patients sitting in front of me, and I may not know their DNA. And in biology and botany, you have important work to do where you may not have the DNA right in front of you. So
exactly. Yeah.
All right. Thank you for your talk. I appreciate it very much.
You're welcome. Thank you. Yes.
Yeah, Karen Sill also says, thank you, Dr. Abbott. This is very helpful and enlightening to her. Rob King says, great job, well explained. So,
Cool.
and Shayna says, thank you. This is so interesting. Yeah, and I looked up those families. I didn't realize they were both the dogwood family. So I just learned something new. The black gums are in the dogwood family.
Historically, yeah, historically they were, but now that's kind of the point. Is
okay.
Nowadays the dogwoods are not being treated as synonymous with the black gums. But I will address why that is the case next week. So.
All right. Well, we look forward to that then. So.
Yep.
All right. Well, thank you, Dr. Abbott. We really appreciate you sharing your expertise with us and teaching us about cladograms and plant phylogeny. This has been very interesting. I want to thank everyone else for being with us here today. Again, the recording of this webinar will be uploaded to our YouTube channel. So go out on YouTube, type in Arkansas Native Plant Society to find it here a little bit later. The next webinar, as Dr. Abbott mentioned, will be next Saturday, where we'll be following up with part 2 to understanding cladograms, where he'll be going through some of the examples that he mentioned, as well as Cornaceae and I know I'll butcher the other name, Nesaceae. You can find out more about the Arkansas Native Plant Society on our website at anps.org. Also, I encourage you to follow us on social media. We are on Facebook. And if you'd like to join, anps.org/join. Thank you very much.